All right, I'd like to extend a thanks to our AV team here at the Dana-Farber Cancer Center Institute. Our next keynote speaker is Dr. Samantha Lent. Dr. Lent did her dissertation work in mathematical methods and correlated data in the laboratory of University. She now works in the diagnostic space and industry. Everybody please give Dr. Samantha Lent a warm welcome and for her next keynote speaker.
All right, let's see if I know how to work any of this technology. We'll find out. Oh, perfect. Okay. So hi, everyone. I am Sam. Today, I'm going to talk to you about weighing the risks and benefits of having quote unquote de-identifiable data in the public sphere. There was actually, I couldn't have asked for a better lead into this than Lauren Harmon gave a talk in the epigenomics session right before this, proposing some solutions. So I'm going to talk about the problem. I was asked to give a patient perspective talk today, and I shoehorned my way into making it more of a technical talk. So coincidentally, I am in the data set that Lauren has been working on, and so I'm going to give my perspective as one of the patients in their research. So some professional disclosures first. I'm a current employee of Freenome, and the reason I bring this up is because this work isn't supported by Freenome and isn't related to my role there. So if you have any questions, comments, complaints, concerns, don't talk to them about this. It's just a hobby project that I do on the side. Some personal disclosures. So today I'm going to be talking a little bit about my research. I'm also going to be talking about my experience as a participant in cancer research. So I want to be really clear that no group is a monolith. And so my perspective as a cancer patient is just that. It's one person's perspective. I think it's interesting to have experienced research from both sides of the coin, but I want it to be something that sparks discussion and doesn't shut it down. I find that when you bring up personal experiences of childhood cancer, it can kind of have a silencing effect, and I really don't want that to happen here. And on a similar note, when you bring up anecdotes about childhood cancer, you hear a lot of like brave and inspiring. So I just want to throw this out there now that I am openly hostile to that kind of thing. So if I hear that directed at me this week, I will spontaneously combust, and you really don't want that blood on your hands. So a little bit about me, how did we get here? So if we flash back to 2008, I was applying to colleges, deciding what I wanted to do with my life. In true teenage fashion, I had the delusional idea that I would become an interpreter despite not yet being fluent in a second foreign language. In 2009, in my spring of my senior year of high school, I was diagnosed with acute myeloid leukemia, so that took up most of the year. And then after treatment, I really didn't want to have this knee-jerk reaction where I said, you know, I survived cancer, and so I'm going to go become a doctor or a nurse or do cancer research. Not that I really even knew what cancer research entailed at that time. So I stayed the course. I went to college for a degree in international relations, and about halfway through hadn't really shaken this urge to get involved in biomedical research. And so I went to lunch with one of my oncologists, and you know, they get to know you pretty well. AML is a pretty intense treatment regimen, so you are inpatient for long periods of time, and I was in school at the time. And so my oncologist kind of said, you know, you really like math, you've always been good at math, have you ever heard of biostatistics? And so the rest is kind of history. A couple weeks later, I changed my major, got my bachelor's in statistics, and then went straight to get my PhD in biostatistics from Boston University. So at BU, I was working on a mix of statistical genetics projects. So I did a little bit of work on genotype imputation, some more statistical genetics work for the Framingham Heart Study. And then on the other side, on my thesis, I did work on methods for analyzing DNA methylation array data. And I had always considered those sort of two separate things. And it wasn't until I graduated and went to the AbbVie Genomics Research Center where they started to come together a little bit. So when I was at the AbbVie Genomics Research Center, I started working on this ancestry prediction tool for methylation array data, which I have presented publicly, so the AbbVie legal team can't come for me for disclosing that. But I was getting some data from Gene Expression Omnibus, and I was standardizing the metadata and realized I had an AML trial. And so I like to keep up on the research, dug into it a little bit more, and pretty easily identified myself in this data set. It was from my clinical trial, and it was my bone marrow sample. So I'm just sitting at my desk one day, and I'm like, oh, hey, there's my tumor data from when I was 17 years old. And so this, you know, I had already been thinking about genetic artifacts and methylation data for this ancestry prediction. We were doing a pretty good job of predicting genetic ancestry. And so I had to kind of have this question lingering in the back of my mind of, you know, how much genetic data can you reconstruct just from public methylation array data? not using any matched phenotype data or anything like that. Unfortunately, it's a little bit hard to get institutional support for that in industry. So when your pitch is, this will not help our company at all, and also I'm trying to make the argument that this public data resource that we're using and getting a lot of mileage from maybe shouldn't exist, it's hard to get buy-in for that. So it's kind of just been on the back burner for a few years. In 2021, I went and did a medical ethics fellowship at the McLean Center for Clinical Medical Ethics at the University of Chicago to try to find a home for that project. Didn't quite work out. So it's just been a thing that I have languishing on my laptop that I kind of pulled together finally for this talk when Tim invited me. So my experience as a research participant, just to give you a sense of sort of the human side of the data you all work with. So within 24 hours of going to the ER, which what I thought was probably a virus, I was diagnosed with AML. I was moved to the University of Chicago for treatment, where I did not come home for 4 weeks, and I received my first dose of chemotherapy as a part of an experimental arm of a clinical trial. So there's a lot that's happening in that first 24 hours, at least for aggressive cancer. And so my clinical trial eventually became part of the Target AML project. So the data I'm going to talk to you about today is actually from that bone marrow sample, within 24 hours of diagnosis. And having done this clinical medical ethics program now, where we talk a lot about the importance of informed consent and revoking informed consent, I will tell you that I have no idea what I've consented to over the last 15 years. I especially don't know what I consented to in those first couple months, right? Like there's a lot happening. You're signing a form. I wouldn't know who to ask about it. In fairness to my treatment team, this is not, their fault, especially in recent years. My response when they ask me about new research projects is kind of like, yeah, fine, whatever, just like give me a paper to sign and move on. But my experience as a participant in research has ranged from recently I had a study of late effects I was involved in, where they sent me the lab results and a manuscript after the fact with all the findings. And so I could actually look at the manuscript, see what they found, and kind of pinpoint myself and say, oh, they found this risk factor, here I am on this plot, here's how this affects me. To, on the other side of the spectrum, accidentally stumbling upon my own data on the internet while I was working. So kind of a full spectrum of experiences. So this Target AML data set that I'll talk about today, this is a pediatric AML study. Like I said, it's come up in a couple talks earlier today in the epigenomics session. And there's extensive omics data available on these samples. And there's also quite a bit of clinical metadata. And so this genomics data commons is a really good resource for the research community. And some of the data is public and some requires DB gap access. So I just put a little screenshot here of sort of the landing page for this project in the genomic data commons. You can see there's about 2,500 cases. It is very user-friendly from a researcher perspective. It's super easy to look up a patient, figure out what kind of data they have, access the data. But you'll notice in the summary where it says, the project has controlled access data, which requires DB gap access. And then they give you instructions. So they've acknowledged that although this is a really valuable tool to the research community, there are some things that should have restricted access. However, the methylation array data from this study is also publicly available on Geo. And so that's what I found while I was looking for public methylation data sets. The only patient metadata that's available on Geo, it's pretty light. It's just the group, so pre-treatment AML or the normal bone marrow samples they included in the study. Age at diagnosis in days, which is how I was pretty easily able to identify myself, and gender. But the title of the samples on Geo contains this target unique subject ID, and so it's pretty easy to then go to the genomic data commons and get access to more extensive metadata. And this has a lot of extensive disease phenotyping and not very much else. So, there are things about the tumor characteristics, there's continuous and categorical measures of residual disease, based on the chemo cycle, there's severity of diagnosis, and some other factors, like how many of your peripheral blood cells had the tumor mutation burden, et cetera. And it was kind of jarring to me because as a researcher, I've had a lot of scenarios where I see a paper and I think, oh, this would be perfect for answering my question. The data is publicly available. And then I go to Geo and it's like, well, here's all the genomics data, but all we have is case control status. And there's nothing I can do with that that's going to add on to what's already been published. right? So I would argue that that's not even really above and beyond like maybe a meta-analysis. It's not that helpful to the research community or to advancing science. So I was pleasantly surprised actually that there was so much clinical metadata available publicly. And I think it's a little counterintuitive that from a patient perspective, more restricted phenotype information is not always better. So if my data is out there publicly, there's some risk of re-identification, there's some privacy risk, but the trade-off there and the reason we say that it's acceptable is that it can further the science and it can help us discover new treatments and all of that. And so if you don't have that upside, then you've exposed risk to patient privacy without contributing anything to the scientific advances, right? And so from my perspective, I think Target AML actually did it really well. And I'm not saying this under duress because Tim invited me. I actually do think they don't have a lot of unnecessary covariates in there, but they have a lot of really good, rich disease phenotyping. So I'm going to dig into the DNA methylation data a little bit more. I'm cognizant of the fact that not everyone here works with this data type, so we'll just do a really high-level overview. And so DNA methylation, it's the addition of a methyl group. Usually you're looking at a CpG dinucleotide or a CpG site, and it's an epigenetic phenomenon, so it's not actually altering your underlying genetic code. And it's tissue specific and it changes over time. So for those reasons, it's usually been seen as less identifiable than genetic variants. And so it's like you're allowed to have samples on Geo where you upload the raw data, and it's treated very differently than uploading, say, a SNP array from the same patient. So we know it's a changing epigenetic feature, but the values are also influenced by genetics. And that can be both via biological mechanisms or via technical artifacts. So the most egregious, there's a bunch of different technical artifacts, and many papers have been published going through them more extensively. But sort of the most egregious one that we point to is a CT SNP underneath the CpG site. So most of the time, the way we measure methylation, whether it's sequencing-based or array-based, will involve bisulfite conversion. And so you have the strand at the top here where 2 of the cytosines are protected by a methyl group and 1 is not. And after you do the bisulfite conversion, the cytosines with the methyl groups attached remain cytosines, and the unmethylated cytosines are converted to uracil. So then in the end, when we do sequencing or PCR, we can look at the T's in the data set and say those are unmethylated C's, or they were originally unmethylated C's. The problem comes with what if you have a genetic mutation at this site? What if you originally had a CT genotype? Then it's not that you have 50% methylation status, it's just that we can't tell the difference between a T allele and an unmethylated C. So we do see examples of this in the literature, actually. And this is an example from a couple years ago, which has ancestry here because it started with this genetic ancestry project. And you can see on the x-axis, these are the individuals, and on the y-axis, this is the beta values, which are generally interpreted as the percent methylation. And the color of the points are those genotypes underneath that cytosine in the CpG site. So as expected, the TT homozygotes have a methylation rate of 0. We can't tell the difference between an unmethylated C and a T. The CC genotypes aren't necessarily at 100%, but we just get their true methylation readout. So we'll have a little bit of variation, but in this case, it's between 80 and 100%. And then those heterozygotes in the red points are right in the middle of that, because half the time we're getting an unmethylated signal from the T, and the other half the time we're getting the true methylation status. And then in addition to these technical artifacts, we can also have genetic information that comes as a consequence of true biological associations. So this is actually hot off the presses this week, and so I just added it because it was convenient. A recent study of 2,000 blood samples had matched phenotype and methylation array data, and they found that methylation levels at over 30% of the CpG sites tested were associated with the genotype of a nearby SNP. And there was a couple of different mechanisms for this. So this is figure 5 from their paper, and you can see at the top, sort of the one we usually expect where they're in the same region and there's this direct influence from a neighboring SNP, but they also found some trans MEQTLs. And what I liked about this paper is they presented the mechanism for some of those MEQTLs and how the SNP would influence methylation levels. So another thing we could look to here is there are public data sources that aggregate these MEQTL signals. And so one of them is this PanCan MEQTL database, and they have MEQTLs for several cancer tissues, including AML samples. So you might think that if you take a bone marrow AML sample, you can look at this database and say, here are all the SNPs that are associated with methylation. Can I kind of backtrack from the methylation status I have to guess what that genotype would have been? So what's the impact of this on patient privacy? One thing I want to distinguish between is a lot of patient privacy research focuses on the risk of re-identification. I've been more concerned about just consistent classification of data rather than the risk of de-identification. But it is important to note that the idea that we need to control access to genetic data while we can have other biological and health data freely available is really a consequence of this genetic exceptionalism. So it's the idea that genetic data is so unique and so private that it needs, special guardrails around it. And I have to acknowledge that it's debatable whether that exceptionalism is reasonable. So there was a paper in 2019 that gets at this, but it is the norm in genomics research. It's the reason that you can upload a methylation array to Geo without any restrictions, but you can't do the same with a SNP array for a patient in your study. So the data I'm using in this project represent a really small portion of the over 120,000 patient samples on Geo that have not just methylation, process methylation array data available, but raw methylation data as well. So the question I had was, how many genotypes can we correctly infer just from a public methylation data set, and how many is too many before we should start considering this genetic data and subject it to this more restricted access level? So there are a couple tools to find SNP-like signals in methylation array data, and like we saw in that plot a few slides ago, you generally see this trimodal pattern where you have clusters of the 3 different genotypes that have similar methylation readouts. And so the original that I'm aware of is Gap Hunter from 2016. And this is a really simple algorithm where you just take all the beta values or the methylation percentages in your data set, order them, and you look for areas where you have huge gaps. You have no one between 20 and 40% methylation. Anyone under 20 is the first cluster, and then you repeat that process. More recently, there's been a little more work on this. So methyl-to-SNP, which uses k-means clustering, and importantly, they add this inverse quantile weighting to identify sites that have 3 equally spaced clusters rather than just 3 clusters. There's a really good one that I just actually found out about in preparing for this talk, Tools, which is using dbScan to define the number of clusters at each site. And that's a limitation right now, is now you have to, if you were to do k-means clustering, you would have to say, I expect 2 clusters at this site or 3 in advance. And so they added this component of looking across the genome and telling you how many clusters was the best fit for the data, and then giving you the cluster output. And then there's also a neglected script on my laptop from 2019, which I'm going to credit to this conference now that it's out in the wild. And this is, I kind of went down a similar path as the Tools people who, had the follow through to actually get theirs published. And so I was using Gaussian mixture models as well on beta values, or k-means clustering on the methylated and unmethylated intensities to try to find sites with 3 clusters. So all of these methods have different arguments that help define what a good cluster is, whether that's how evenly spaced they are, how many people you need in a cluster to think it's a true association rather than just an outlier, and the probability of cluster assignment. I'm not going to get into the details of the arguments I use for all of these. I tried to make them as comparable as possible, but I am always happy to talk about this stuff. If anyone has any questions, we can dig into it. So I applied these tools to the target AML data set. And again, this includes AML samples and also healthy bone marrow. Cancer genomes are weird and they are varied. And so I restricted to just things, and it'll be clear later on why I needed myself in this data set, but I restricted to things that have the same mutation type that I did in my AML sample. So there were 41 healthy bone marrow samples and 61 AML samples with this 821 translocation. And for the clustering methods, I restricted to sites that had an observed beta value range greater than 40%. And this is philosophically a sound idea, because if you're trying to find things that have a 10% gap between them, you obviously aren't going to find that if your data set ranges from 0% methylation to 5% methylation. But also, again, I want to emphasize this is something I do for fun on my laptop without a computing cluster. So we got to get rid of the things that are just a waste of time. So this left 23,000 sites in the control data set and 246,000 sites in the case data set. And you can see here from these histograms, these are the range of methylation values or the spread of methylation in the data. And the AML, like even after restricting to the same tumor characteristics, are just like way more variable than the healthy bone marrow data. So when I applied all of these methods to try to find these SNP-like signals, these three different clusters of points, what I found was that many, like in all of the methods, they found many more SNP-like signals in the AML samples than they did in the healthy bone marrow samples. And again, we tested a lot more sites. We had a lot more sites that passed that, minimum of 40% range across the data criteria. But also, again, cancer samples are really weird. And I think that's the real cause for this, and we'll see in a second. I don't trust these calls from the AML data as much as I trust them from the healthy bone marrow. And you'll notice that generally we have between a few hundred to like just over 1,000 SNP-like signals that we've identified, except for my one at the bottom, my implementation of GMM, which I have clearly not calibrated well because it has a suspiciously high number of SNP-like signals. So I wanted to give it a chance. Like I was willing to say, okay, maybe this is more sensitive. Probably I messed something up. But let's just like look at the things that GMM identified that nothing else did. And so since it's technically possible that it was doing a good job, I looked at some of the individual sites, and I just wanted to show you here what these patterns look like in the data. So on the top, we have a CPG site that has what I would call a really clean SNP signal. It was identified by multiple methods. So whether you visualize it as the methylated intensity against the unmethylated intensity, or if you visualize it on this beta or percent methylation scale, it looks like you're looking at GWAS array data, right? Like you see these very clear 3 clouds of points that you could assign to a genotype. If we look at the bottom, the signal identified by GMM, which we were applying to the beta values, and the beta values, you can kind of see where it was coming from with the AML. Like you could maybe draw some lines there and get 3 clusters, but then when you look at the methylated and unmethylated intensities, like there's nothing going on there. I don't trust that at all. So my takeaway from this was that I need to go back and revisit some of my GMM criteria, and so we just removed it from the following slides. So of the remaining methods, I wanted to look at the agreement in which sites did they detect that had these SNP-like patterns. And there's much more agreement between methods when you're analyzing the healthy bone marrow than when you're analyzing the AML samples. So on the left, we have an offset plot for the AML samples. And so that histogram is just the number of CPG sites, and that lower panel just shows you which methods identified that CPG site as potentially having a genetic artifact. And the top 3 most frequent categories just come from individual methods, and they weren't found by another one. So that looks a little suspicious to me. Versus when you look at the right plot, the SNP-like signals in the healthy bone marrow, now we start to see more agreement between methods. Gap Hunter definitely identified a ton of sites that weren't found by any other method, but the next most frequent category, we had consensus across 3 different tools. So I feel much more confident in those. The coloring of this plot is actually CPG sites in green have not been previously identified. There was a paper in 2016 that put out a bunch of predicted SNP artifacts, so SNPs that lied either right underneath the CPG site, next to it, or somewhere on the probe. And so we would expect that we know some of these have, not all of them necessarily, but some of them will impact methylation. So we'd expect that our results should be enriched for those sites. And we don't see that happening in the AML samples, but we do see that most of the SNP-like signals we're picking up on in healthy bone marrow have been previously annotated to have potential genetic artifacts. So I have a lot more faith in applying these methods to healthy data than I do cancer methylation data. But now we just have methylation clusters, right? Like you can give me a methylation cluster, I still haven't identified a genotype. And so it would be super straightforward to go from a methylation cluster to a genotype if you had matched methylation and genotype data in the relevant tissue. I, again, don't have that because I'm doing this for fun and I don't have an institutional affiliation. But also we want to try it the hard way and like actually keep ourselves honest. What can we recreate just from public data? So we have 2 options without needing individual genetic data. We can use the healthy bone marrow data and just public allele frequencies from that McCartney paper to guess which allele is associated with higher methylation. Just compare our cluster counts to the minor allele frequency and see, do we think we're looking at the SNP that was annotated to this probe by McCartney et The other option is we can use the AML data and the PanCan-MEQTL database to guess which allele is associated with higher methylation based on the effect sizes. I opted not to go that route just because, again, I trust those healthy bone marrow signals a little bit more. And also, there's just a lot more SNP annotations. Like that PanCan-MEQTL is a maximum of 11,000 sites, and that's out of an array of 850,000. So it's a very small proportion of the array that's annotated to have AML-MEQTLs. So I just, for time's sake, only did this for the gap hunter clustering results. So I went through, I compared, I guessed what the allele frequency was in my data with 41 patients, and I compared it to this McCartney data and just tried to get a sense of, do I think that I'm looking at the SNP I think I'm looking at? And what that looks like is a little bit messy. But we estimate that minor allele frequency in our data set, and then when we compare it, we see that we have the best concordance. So McCartney et al. Split up their reported allele frequencies by population. So we compare our estimated frequency with each of these populations separately. And we actually, in the fourth plot there, have pretty good concordance overall with the estimated allele frequencies. So there's a pretty high correlation with the annotated allele frequencies and the ones we're estimating. We do see this big line on the left there, which has a minor allele frequency of 0 or near 0 in the McCartney paper and seems to be very common in our results. This could be MEQTLs, right? This could be something that's not just a SNP on the probe causing problems, but actually a biological association. And we're not dealing with those for now, but we will hopefully deal with them in the future. So now, all we need, we have one geodata set, and we have allele frequencies from a single supplementary table. And I would argue that this gives us all the pieces we need to guess some of my genotypes. So we start with using Gap Hunter to find 3 distinct clusters. And to train this data to look for the gaps, I put all the healthy bone marrow samples in there and just the one AML sample that we were interested in. So this gives us about 2,000 sites to start with that we think might have genetic artifacts. I removed what I'm calling uncommon SNPs, and this is just, we are dealing with a really limited sample size here, right? So anything less than a minor allele frequency of about 0.2, we just, with 42 patients, are likely not going to observe more than one homozygote with a minor allele. So we're really limited by our sample size here. So I removed the ones that I thought were too rare to be appropriate for this sample size, and also the SNPs with a minor allele frequency greater than 0.4, because at that point, there's so much uncertainty in the estimate that we just can't confidently guess which one's the minor allele and which is the major allele. So at this point, we're down to 588 sites, quickly dwindling. And then I looked back at those plots from the previous, and I said, where are the 2 allele frequencies generally concordant? So if they disagree by more than 0.1, I'm going to remove them from the data set and say, hey, the SNP that was annotated to the probe probably isn't the one we're looking at here based on our data. And then finally restricted to biallelic SNPs. So this left us with 463 sites. So a very small proportion of this methylation array, but also this is like some pretty strict filtering criteria. It was really just a proof of concept of what can we call that we're confident. And then, because I have my 23andMe data, and that's the only genetic data I have access to right now, I went in and I looked at the called genotypes that I have. So I now have 463 genotypes that I called with an associated SNP ID in my AML sample. And of these 463 SNPs, 52 were present in my 23andMe data, and I got 51 of them right. So we have 98% of the genotypes were called correctly from methylation array data and population allele frequencies alone. There's no reason to think that the 23andMe SNPs are any different than the ones that weren't on 23andMe. So if we assume that this accuracy rate holds for SNPs not included in the 23andMe array, this would mean that we correctly inferred about 450 SNP genotypes without ever using any individual genotype data. So the conclusions from this, there is some hidden genetic data in methylation arrays, even when you have a funky genome like a cancer sample. And this is just going to get worse as the density of arrays increases, right? But also as computational methods advance. And if you believe in this genetic exceptionalism, again, I think we can debate, this raises the question of how many SNPs is too many before we really need to be treating this as genetic data, or to get back to Lauren's presentation earlier, before we have to create some tools to at least, when we're putting this data out in the public sphere, kind of mask some of these more identifying sites. And maybe counterintuitively, I want to emphasize how easy this analysis was and how much room for improvement there was. This is a very limited attempt to infer genotypes, right? Like this is something that I just did on my laptop with a really small data set. And there are some really obvious improvements that I think would lead to recovery of more genotypes. Um, First, adding the SNP signals detected by other methods. I just used the gap hunter results for this. Another thing would be imputation of the called genotypes. So if you get some neighboring SNPs together, we can maybe use an imputation pipeline to fill in the rest of the region. Using a larger data set so we can detect clusters at much lower minor allele frequencies. And then also, I think there's a lot of work to be done about the filtering criteria, testing different thresholds of how we're filtering things like the allele frequency matching, and seeing how much accuracy we sacrifice as we get more and more permissive with those thresholds. And then I think the last one is to get access to a computing cluster again. So if you want to collaborate, you can email me. My email is up there. And that is it for me. So does anyone have any questions? Oh, we have one back there. Oh, am I delivering?
Hi, very interesting research. I had a question. I think the current most widely available methylation array, like EPIC, for example, already has a few SNP probes that are incorporated. I think 70 or 80 or so. Have you tried using them instead of using SNP-like probes?
Yeah, so there are I think there's 59 probes on the EPIC array that are meant to identify sample swaps. Most normalization tools are going to automatically filter those out. So in this analysis, I just restricted to the CPG sites. But yes, that would be an obvious, again, area for improvement where we just throw in those SNPs that are actually intentionally put on the array.
So thinking of like this, and I must think like even we'll say with RNA-seq, where if we've got the BAM files, we can call SNPs on this. There's a lot of data up there where you can either infer SNPs or, or where you can, where they are actually available, you don't even need to search. What do you think the solution is? Should more data be protected, or is it a case we should take kind of George Church's view, which is there's protection, something that we're just going to have to live with, that we can't do?
Yeah, I mean, it's always going to be impossible to totally protect everything, right? So like, I think we have to have a conversation about how much genetic data is too much. And the problem is that's going to change, right? As computational methods evolve, we are going to be able to uncover more and more. And so there's a question of, well, who's in charge of, this data has been in the public sphere for 10 years and someone forgot about it because the grad student who uploaded it moved on and has another job now, is a little bit tricky. But I don't think we should be making that decision on people's behalf. I think I'm a little skeptical of the idea of this genetic exceptionalism. I think it needs to be about the scale of the data, but it is ultimately something we should put in consent forms, right? It's widely accepted enough that genetic data has this special sort of classification that if we're planning on uploading epigenetic data, maybe we should just be putting in the consent form, do you consent to have your genetic data freely shared before we upload it to a public space?
And this was the second question then was, related to kind of the differences between global interpretation of data protection, we say GDPR versus the US, where there's this right to be forgotten and within GDPR legislation. And I think, there's similar legislation, I think in Brazil and maybe a couple of other countries. So, will we be facing a situation in like not too distant future where people are saying, well, actually, please remove my data, and we have those protections put into the data in the first place?
So you're making me really wish that I had read the paper on the GDPR that I saw a couple weeks ago and never got around to reading. But specifically about this, like genetic consent and GDPR. How do you opt out? I mean, like from a patient perspective, I don't know how well, now I know who's analyzing it, so I guess I can take it at the chin there. But 5 years ago, I had no idea that it existed, much less how to opt out of it, right? So I think there are very practical constraints about how do patients know which data sets they're in, and how would you even go about revoking consent for that?
Final question, I'm sorry, which is, you mentioned earlier on about like the two different sides of the scale of being a patient involved, where you didn't, find the data public, or the other side where you were given the paper. Does that then open up where, how should we as researchers think about, should patients get a copy of every paper in which their data is actually used?
Yeah, so it depends on the study design, right? So I think we've shifted really towards broad genetic consent, which is just, I give genetic consent. I accept at the start of this study that you cannot predict exactly what projects are going to come from it. And I trust the institution to use their discretion. So that's like for biobank data, right? We can't predict when you enroll in UK Biobank exactly what paper is going to come out. It would also be incredibly it would be impossible, right, to circulate every single UK Biobank paper to every single UK Biobank participant. So I think there are studies where it's appropriate. So the study where the results came back to me, it was a very focused, like they were collecting dried blood spots from a particular set of patients. It was unlikely to be reused. I don't think that is feasible for things like target AML. So in an ideal world, yeah, you'd know where your data was and you'd know the results of it, but I really don't know how we go about executing that in any sort of large scale study, unfortunately.
I'd like to thank Dr. Lent yet again for providing perspective, probably close to unique, if not, maybe not completely unique, but certainly articulated in a way that's not a perspective that an awful lot of us would necessarily have. So,
All right, thank you all.
Thanks everybody else.